My name is Marco Guerrini. I'm the Vice Director of the Ronzoni Institute. The last 40 years, under the leadership of Professor Casu, our attention was focused mainly on glycosaminoglycans, particularly heparin and heparan sulfate. Heparin is a well-known anticoagulant drug and used extensively from the 70 years ago in the clinics. The complex structure of these molecules, this mixture of differently sulfated polysaccharide, is very challenging. And the correlation between the structure and the different biological activity that this molecule has are still not fully disclosed. Until 10 years, 15 years ago, the use of NMR was mostly dedicated to the research side, to study conformation and interaction between these molecules with protein or characterization of specific sequences. The situation changed in 2007-2008 after the so-called heparin crisis. The heparin crisis was due to contamination of some batches of heparin, and this caused the death of dozens of people in the US and thousands of serious side effects for patients that were treated with contaminated samples. The heparin crisis forced agency, FDA and EDQM in Europe, to introduce new tests to better control the quality of heparin. At that point, the proton NMR was included in the pharmacopoeia as a test for drug release. Even though the proton NMR can help in determination of purity of a sample, it cannot provide a full description of the composition and characterization of the product. About 10 years ago, we started to use 2D NMR, particularly HSQC experiments, because this experiment allowed to spread signal in 2 dimensions, so we have much more information about the composition of heparin. And we try to use these signals and to treat these signals in a quantitative way, so to determine the relative composition of a heparin, of each batch of heparin. Thanks to the collaboration with the Food and Drug Administration analytical group of St. Louis, we optimized the method, investigating in all parameters that can affect the results. So we verify, we demonstrate that this method is reproducible, is precise, and can be used to compare batch to batch in industrial production or identify possible contaminant that can be present in the product. After development of this quantitative HSQC method, we realized that this application is limited to very well-skilled lab, NMR lab, but we would like to give a possibility to other groups and other labs to use the same methodology. Thanks to the historical collaboration that the Institute has with Bruker, we decided to implement and to try to implement the method on AssureNMR. AssureNMR can help us because it's possible to automatize all operations that manually are done during the analysis in a fully automatic way. So there is not anymore in the influence of operator, and all operation from measurement, calibration, measurement, and elaboration of data can be done automatically. One of the key benefits of AssureNMR is that this method is fully standardized and can be used in GMP environment. For this reason, we use already use Assure to run NMR tests present in pharmacopoeia. Even though this method, the quantitative HSQC method, appear quite complex, I believe that thanks to AssureNMR, it could be implemented in the pharmacopoeia as a routinary test to heparin and complex drug molecules control.